Hi, this is Dr. Ben Morrill. Welcome to episode 63 of Reptile Genetics Weekly. Glad to have you here and hope to see many of you this next weekend at the Pomona Show. We'll talk a little bit about that later on. The raffle we'll be doing and get together, all the stuff we got going on for Pomona. But yeah, in the meantime, as far as results going out, I'll probably stop announcing it since it's been consistent so long. With fast tests are going out almost every day. And then sex determination for ball python and clubrids is still usually we're running them twice a week. So it's usually 3 to 5 business days from when we get them. And the fast tests are just 1 to 2 business days. And then the panels, we had a little bit of a setback for the current ones we're running. And the genomics center that runs those for us had some things pop up and had to fix a machine. So anyway, that's a little delayed. I'm still hoping that we will be able to get results by the Pomona Show. So it might be like the day of that I'll have to be analyzing like crazy, or maybe even the morning of or something. But we'll try to get as much of that and as much of those results out as we can. For the panels, and if we can't get them out before the Pomona Show, then we will get them out as quick as we can on Monday, Tuesday after the Pomona Show this weekend. So it's coming. Unfortunately, when the machines are working great, everything's beautiful, but sometimes there's a hiccup and we hit one of those. So but yeah, that's what I have as far as samples. Kayla, how are you doing?
Hey Ben, I'm doing well. Let's see, I got really good pictures of some of my babies. One of them was in shed, but happiness is a fistful of baby snakes. And so life's good. Everybody's eating whether or not they like it. This one's still being assist fed, but the little dude who had a kink in his spine, we've nicknamed him Ruffles because he has ridges. Little Ruffles is eating and pooping well. So looks like the kink is just a cosmetic hiccup, and otherwise he'll make a great pet only animal for somebody. So yay.
We were talking before, I already told you this, but I love those eyes. Those eyes look awesome. So
right. And there's a chance that Ruffles is actually going to be a female. I guess we'll find out whenever I get the results back. So but it seems like in the lightning rat snakes, the females, at least from what I've noticed between me and some of the other breeders, it's a newer morph too. So, but it looks like the females might actually have a higher intensity in their reds and coloration compared to the males. So we'll see. That might just be, may or may not be a thing, but even still, real pretty. So
yeah, sorry, Kayla. Kayla gave her sheds to me since we live close, and I'm not as fast as Texas. So
oh no, that's totally fine. I don't mind at all.
I'll get them to you, I promise.
All good. Take your time. I mean, we kind of have a business to run too, so that's okay.
Yeah, it happens.
Yeah. Speaking of business, we do have some sales going on this month with our partners, MorphMarket and Clutch. So be sure to check those out. We're going to mention these each week it's going on. And I guess we have 6 days left for, not for this one. Clutch is
this one's ongoing.
Yeah.
This one you'll be able to do on Clutch anytime from now on. So they have it kind of like, this was actually an idea that they had at Clutch to have these bundles, which is kind of similar to what we did with the Pick 4, but this makes it completely customizable. You can do 2, you can do 3, 4, 5, 6, 7, however many you want, and just choose what size of a bundle. So if there's, an animal you want to know 5 mutations on, and you can pay for a 5 bundle and get those, and those will all be fast tests. So whether you do 3 or 7 or whatever, they're going to all be fast tests. And after you pick up like a 5 test is $110. And after that, it's just another $10 for each additional test. And so it's a pretty good way, pretty sweet discount if you have several, especially if you have several animals that you want to know multiple genes for. I know a lot of you are going for quad hets and stuff like that. This is a pretty sweet way to do that.
Yeah, I get you some tasty bundled shed tests a la carte overnight. That should be the marketing for it. And then MorphMarket. Then this is the one that's through the end of June. That's 25% off for everything. Shed test wise.
Yeah. So jump on it. Yeah, jump on it while you can, because I don't know for sure if we'll have, now that we're getting to where we're getting into baby season, we're getting more shed samples coming in. I don't know how many more deals or how good of deals we're going to have. So jump on it while you can, because I literally don't know for sure what will come after June. So
Yeah. No, I do the social media. Ben does the science. Sean and MorphMarket just kind of do what they want to do, and we get to tell you. So, so jump on that while you can. And of course, with baby season here, you can order in bulk. If you're ordering like 25 or more shed tests, send us an email at raregeninc. The words are hard. raregeninc@gmail. So we'll post it everywhere. So if you got those questions, let's see if we can make you an even better discount for those bulk orders. So we'd like to hook you guys up if we can.
Yeah, definitely no problem paying for them now at a discount, and then you can through the year. That's no problem at all. Prepay, pay for it now at a discount. Use it in August and September. Totally fine.
Yep. Awesome. So you and Sean are going to the Pomona, California show.
Yes, excited to be in California, Southern California again, and see many of you that I got to see last year. Sean will be the first time he'll get to meet a lot of you in person, and I'm sure we'll meet several of you that I didn't get to meet last year. So looking forward to that. We should be fairly close to Dark Cloud Reptiles. It will either be right next to them or very close to. So those of you that found us last year will be in a similar spot. Yeah,
definitely. And so can we bring sheds to you?
Yeah. So bring your sheds. If you've got some saved up, some you've, wanted to do for a long time, bring them. The easiest, simplest way is to go ahead and pay for them before you come and have the bags marked with your order information. But if you don't, you want to bring them and, just pay for them there. We'll figure that out too. So just bring your sheds, and any of you that have some, like for a really dark axanthic, we've been trying to collect sheds for really dark axanthics. For any of the tests or any of the morphs we don't have a test for yet that you want to help with development, bring those as well.
Awesome. What if I have sheds for a species that there isn't testing for yet, but it's going to be for like research and stuff? Can I bring those too?
Oh yes. Yeah. All that stuff. I'll happily take bags and bags of sheds home, add them to the collection, and put them to work.
Awesome. Boa breeders, get on it.
That's right.
And keepers, anything really, but we're on the hunt for a few specific things, and we're looking for boa sheds too. So
boas and hogs and corns, that we're starting to get a decent amount of all three of those, and hope to start working in those pretty soon. So if you have any of those are really good ones to get into us. But even if you have other species, we'll take them, and Kayla's been working on organizing, and we'll have them organized. And as soon as we have enough of something, we can run with it. So exactly.
All right. And we do have our big kid tarp or setup, not a tarp, but what's the form? I don't know what the formal term for that is, but we have our branding. It'll be much easier to see. So look for Randy and look for this, and you'll find us.
Have actual real vending stuff, better than we did last year.
Yeah, it even comes with a Sean next to it, most likely. Although Sean tends to run around and he hunts people down to find them and then talk to them at the shows. Like, he is nonstop. So I'm sure you'll find him. You just don't know where.
It might be the only time you see him by this banner.
Exactly. But there's also the pre-show. Go get a beer before the show. And here, Ben, if you want to talk about that.
Yeah, free beer, definitely. Come and say hi, talk reptiles. Just when you show up at Tortuga Brewing, let them know you're with the RGI group, and come talk reptiles, and get prepared for the show with some free beer. So
yeah, there's a food vendor there too. It's at Yakitori Cartel if you want to check out what they cook. I've looked them up on Instagram and it looks really good. So definitely go check it out. Sad I'm missing it this year. Hoping I get to make it next year. So definitely don't miss out. If you're going to Pomona, let us know. We'd love to see you there at the pre-show party.
And we mentioned it before when Joe was on real brief, but this is a family establishment. So if you have kids with you, that's totally fine. He's done birthday parties there. So yeah, you're good. It'll be a family friendly atmosphere as well.
Definitely. Yeah, that's really important too. So if the kids are coming along, that's totally fine. Let's see. So we have ironed out some details on the raffle that we're going to have at this event. Last year we had a really nice puzzle snake to raffle out, and this time it is a male Cypress Lesser Pastel Hitari, and he is breeding size, ready to go. And he was also donated for this raffle by our friend Randy at Dark Cloud Reptiles. And he's so sweet, you guys. It's so pretty in person too. Like, he's unbelievable.
Yes, very thankful to Randy. He's excited to be able to get a Hitari test going, and him and Brittany Gobble have sent in quite a few Hitari and Atari sheds.
Mm-hmm.
And so this will help fund that, be able to make that test available. And any of you that know Randy, give him a shout out, or say hi, shoot him a text or whatever.
Yeah.
He let me know he's had some stuff in his personal life, and I won't give details, but he's having a rougher week than he was expecting. So let him know you're thinking of him, and I'm sure he'll still be at the show, but he's having a rougher week than he thought he was going to have. So thinking of you, Randy, and excited to do this raffle and get this Atari test going.
Yeah, we love you, man. Stay strong. So that's the first place prize for the raffle that we have. We've also added a second place prize, which is 2 30 morph panel tests, which is a value of, what did we say that was? About $260 worth of testing. That's huge.
So it's essentially 60 tests, 30 for one animal and 30 for another. So
yeah, and that's just the second place prize. And then the other one, third place prize, is 2 single gene tests for ball pythons. If you have colubrids or something and you'd prefer that, just shoot us a message and we can work something out that way too. But either way, they're really nice prizes, and all of this is in one raffle. So one raffle ticket has a chance at any of these.
And Kayla will be working on that specific raffle, getting it set up and at the show. So you'll be able to enter, pay for raffle tickets and enter online. Or if you come to the show, we'll have a sheet. Any of you that did it last year will have the same thing. Kayla's working that out. So we'll have a sheet there. You can just scan it with your phone and pay for however many raffle tickets you want. And then at the specified time, I think we did it at 2 or 3 o'clock on Sunday. Kayla will do the draw. She'll hit the number generator and let us know who the winner is. And maybe we'll even figure out if we can get it on a laptop so people can watch you do it this time, but we'll see.
Yeah, we'll see what we can do. But either way, it's something that you can participate at the show or online, even if you can't be there. So everybody's got a shot at it. And honestly, it's a really, really sweet deal. So definitely don't want to miss it. So keep an eye on our social media.
And all that money goes directly to helping make this Hitari test possible. So that'll make Randy and Brittany especially happy, and several other people that either already have Hitari animals or are looking to get some.
Yep. And plus, if you win the Hitari animal, then you're already in on the project. So
boom,
that would be cool. So last week we had we showed off this little what would we call it? Kind of a guessing game.
Yeah. It was a really interesting pairing that had many different ways to make, either all white or essentially animals that would be really difficult to pick out by eye. And when I came across it on Facebook, I was like, okay, we got to test these and do what we can. So of the 6 different morphs in these, potentially could be in these animals, there's 3 that we could test right now. So we ran 3 tests on them. We did a giveaway, which we'll share the results. But then down the road, when we can test the other 3, we'll bring these animals up again and see how y'all did guessing the other 3.
Yeah. I love that. This would have, before genetic testing, this would have been a nightmare to sort through. I mean, they're gorgeous animals, but it's like, okay, we have banana bamboo leopard Enchi fire crossed with a banana bamboo champagne possible leopard Enchi fire. It's like, okay, here's 5 white snakes. Have fun. But now we can prove out what it is, or at least most of it, and the rest we'll be able to prove out later once we have those tests. So Here are 5 white snakes. I really love number 5. That's so pretty. And I think I just like the water droplets on there too. Looks nice with the steel blue eyes. But the guy who guessed the closest was Will Adams on Facebook. And the results came back as, well, there you go. 4 of them were super bamboos.
Yeah, crazy.
Like, what crazy odds.
Yeah.
Yeah, and no leopards at all, which surprised everybody.
Whiffed on the leopards. Got more than half inchies. But yeah, those bamboo results were crazy when I saw that. Because we don't, honestly, in testing, we don't see very many. I have run a few super bamboos, but I've never done a run and had 4 super bamboos in that run. That's a lot of super bamboos.
Yes, it is. And I think we were talking about how, like, we were suspecting leopard in some of them because of, like, you said something about the head scales.
Yeah.
But, and, you can kind of see, like, some funny head scale things going on in these photos, but nope, not a single leopard in this bunch. So that is coconuts. But definitely really fun that we can do this sort of thing. So I mean, if y'all find any other like crazy pairing examples like this, let us know. Like, tag us on whatever social media you're on, because this was fun. And of course, Will Adams won some free genetic testing from us for it, for getting the best guess.
Yep, he already reached out. He's got a Bang Pos het pied girl he's going to send in for a het pied test. So.
All right. Well, let's hope for a het pied for him. That was, fingers crossed. So now for, I guess, well, I mean, shoot, we've covered a lot, but I guess this is the actual meat and potatoes of this episode. And that is our monthly-ish Morph Spotlight, this time for Spark and Specter. Almost done with the yellow belly complex, because we're kind of moving in the order that we that these tests were designed. So let's get started.
All right. So here's a killer looking one. I think this has got yellow belly and then either gravel or spark.
I think this one was
or it is a gravel and it is either spark or specter, one of those two. But yeah. So Ryan with Aesthetic Selection Reptiles, he's done some really cool stuff with Specter, especially in clown. Super Specters. But yeah, so really cool animal. Want to give him a shout out. He's done some cool stuff with the kind of lesser used yellow belly complex genes.
Yeah, they're really pretty. So let's talk about how it works. So firstly, like our previous yellow belly complex, this comes from the same paper. And Ben, did you want to take it away from here? Yeah.
So kind of the two different things that we're seeing associated with traits in the mutations in the yellow belly complex that are in this endothelin gene is making it leucistic or all white, and then also with striping. And specifically with Specter and Spark, they're both single nucleotide polymorphism. That's what SNP stands for. So with yellow belly, we had a deletion. We had a -1 or a -17. Right. With both Specter and Spark, there's just like one A, T, C, or G that just like say it was a T and a normal and it changed to a G. It's just one little simple change. And that's what makes it a single nucleotide polymorphism or a SNP. So if you hear genetics people talk, a lot of the time we'll talk about a SNP mutation. That's just one of those bases changing from being a T to a C or a G to an A or whatever. Okay. And so both Specter and Spark are just simple base changes. So there's not even a change in length like there was with yellow belly. It's just simply one base changing.
Neat. One of the visuals that they had in the paper, and I really like this because it shows, here, let me remove that for you. It shows what we see as breeders, or what's described as breeders. And here they explain like the phenotypic severity. So the gene like yellow belly and those mutations have the most severe impressions. Right below that, we have Spark and Specter. And then a less severe change in mutation being gravel and asphalt. That we'll talk about next week. And the stuff on the far right that says frame shift and missense and G to A substitution, don't be afraid. We'll talk about that in a future episode. We're going to cover it, so just focus on the left side for now.
Yeah.
Do you want to add anything to that, Ben?
Just very cool that they were able to figure out these very specific details and be able to show how the kind of mutation. So we, as keepers and breeders, just talk about mutations, but there are different kinds of mutations. And specifically with the yellow belly complex, the different kinds of mutations make it display a different level of severity, is how they say it. So how much lightning there is and how much striping there is, that puts Spark and Specter kind of in the middle. And yellow belly is really powerful because it makes it all white. Then gravel and asphalt, you don't even get that perfect stripe or anything. So it's kind of the least severe of the mutations. So very, very cool paper. And if you want to take a little time to check it out, I know there's going to be lots of big words and sciencey stuff, but it's kind of cool just to see, because it's actually about the animals that we keep and love,
Yeah. Plus, I've noticed, as somebody who doesn't, as someone who is more of a layman than, say, Ben, who is the geneticist, sometimes just reading those papers, even if I don't fully understand each one, like just kind of absorbing that information, letting it digest. I found that when I hear Ben talk about things later, I'll be like, oh, I know that word. And, sometimes I'll like Google it and be like, okay, I'm starting to put some pieces together. So there are ways to make it more accessible over time, but we're also working on episodes in the future to make it less scary. So
we'll have to get Travis on to help give you some good examples and ways to think about it.
Definitely. Yeah, that'll be fun. I like, he has a lot of good, easier ways of explaining more complex subjects in genetics. So that'll be cool. And this, you said these are the
so these the blocks. So this is the genetic code. Like if you this is kind of like the 10,000 foot view of it. You're zoomed out enough that you can't see the A's, T's, C's, and G's.
Okay.
But the blocks, those are the parts of the DNA code that actually code for the protein. Those are called exons. And then the sequence in between, those are called introns. So the lines in between, that sequence doesn't specifically code for amino acids that will be part of the protein, that endothelin protein. It's just those blocks, those exons. So that first exon for the endothelin gene, that L152F, in a Spark, there's that one base change that changes one amino acid. And then the next one down in Specter, you can see it's 1, 2, 3, 4, then the fifth exon there, that we have another base change that causes a amino acid change also from an R to a P. And that's what makes it look like a Specter.
Okay. And so those are the codes, those codes specifically for the proteins. And here in these two figures from the paper as well, show what those like the shape of those proteins. And if I blow it up a little bit easier, you'll see on the left side, you see the Spark, and that one little cyan spot is the protein that that helps create what the Spark phenotype.
Yeah, so that's showing you the specific amino acid that gets changed, and so
that's it, yeah.
that those two horizontal lines, that's showing you the cell membrane. So this is a protein that's embedded in the cell membrane, where part of the protein's on the outside of the membrane, and part of the protein's inside. And that L to F change at amino acid 152, that's like actually inside the cell membrane.
Oh, cool.
Normally. And then with Specter, it's a little different. It's part of the protein that's a loop that's outside of the membrane, and that's an R to a P amino acid change. So the protein looks similar in shape for the Spark protein and the Specter protein, but those little changes actually make a difference with how the protein interacts with other things, and that's why it makes the animal in the end look a little different.
That is amazing that such a tiny change can create this visual effect that we as breeders describe as the genes that we throw up on MorphMarket. And so that entire thing is created by just that little change in the so correct me. Okay, let me make sure, because I'm learning this as the audience is as well. So that is the protein in along the cell membrane of
Yeah.
each cell of this animal.
Yep.
Cool.
Yeah.
That's awesome. Well, neat. Let us know if you guys like this sort of thing, because my nerdy side adores it. If this is something that you like to see as snake breeders, and like, if you work with these genes, being like, oh my gosh, that's you holding up your ball python, I think it's great. So let us know what you think. Of course, here are the folks who contributed animals to, sheds for mapping the yellow belly complex. Lots of familiar faces and, names, I suppose. And so how well established is this test, or these tests?
Yeah, so for both Spark and Specter, since they're part of the publication that Dr. Hannah Seidel did along with her students and collaborators, so that makes it possible to be an A+, and it is an A+ for both of them, because we've run a bunch, Dr. Seidel's run a bunch, Gray Rider Reptiles, they use these same tests, Dr. Rafi in Canada, people in Europe, like we all use these same tests, because Hannah did such a great job with this publication of finding out very specifically what these changes are. So they're very, very good tests in the yellow belly complex.
That's awesome. All right. And so now it's time for our shed donor shout out. This time it is Snake Ltd. And he has, I don't know if you can, let me blow this up bigger for you to see. He has some really cool sales going on. The mail sale, I think, will be over like shortly after this video publishes, but he posts stuff like that. Has some really good looking animals. What did he donate a bunch of sheds for, Monarch?
He's got a bunch of MJ Xanthic stuff.
That's it.
Yeah. So he sent quite a bit of that, and he's run other things with us as well. But yeah, that's what he's sent in most recently. And as soon as that MJ/GCR test is working right on, I'll run the rest of them for you. I know he's waiting for me to get them all run, and until we have a test that we know for sure is working right, I don't want to run too many things when I can't say for sure if it's correct or not.
Yeah, of course. Makes sense. Well, cool. Definitely go check out his stuff. I'll have a link to his MorphMarket page in the description of this video. And thank you, Snake Ltd, for your contributions to our work. And I think that's actually everything we have for this episode, Ben. So you got anything else for us before we call it a day?
No, just come see us at Pomona if you're going to be there. Come to the Friday night get together and get some good conversation and free beer if you want. Definitely looking forward to that. That'll be a lot of fun. And yeah, bring sheds. That's really all that I've got. I'm excited to see y'all.
Awesome. Well, sounds good. And everybody travel safely on your way to and from Pomona. And I guess we'll see you guys next week. All right. Now, outro in 3, 2, 1.